Okay, yeah, thanks everyone. Thanks for the few who made it today. Maybe a lot of people are out for vacation. But yeah, so there's some news. Finally, this paper has been accepted. I think a lot of you, some of you are part of this. It'll be out next week. So that was finally out. There's other papers in the works, I think people are working on. Uh, a newer thing that maybe some of you might be interested in getting involved with, where me, Paco, and some other people are going to start thinking of organizing a meeting for next summer in person, talking about infectious diseases and covering everything to space. So it's going to be a smaller size meeting, and basically going to be focused on regulatory RNAs and this kind of the topic that we're focusing on probably. So people know sponsors who might want to sponsor. We're thinking of Lisbon around this time. That date's still flexible, probably, but it's going to be around this time. It's going to be 3 days. I imagine some of you here might be interested in participating, or if you guys know sponsors or potential European colleagues or places, I might reach out to a few folks within the group too to see if they want to help organize the meeting. But maybe tentatively put that for sometime in July, if we're able to make it happen. But we'll see. No new updates, but let's just go into the news. We've been hearing about hospitalizations rising, but again, it's a minor rise of 5% increase. And we know this from the wastewater is going up. So the good news is that, the hospitalization, even though it's increasing, is still relatively low, and severe illness is relatively low. So related to that, we know that we just heard that potentially CDC will approve that by end of this month or next month potentially, the new cocktail for the COVID boosters will be available that would have XBB.1.5. But the unfortunate thing is XBB, as we know, .1.5 is going out, and there's that new variant we talked about last week, the EG, which is coming. So I'm not sure how effective. I'm sure, boosters are going to be effective regardless, but targeting the newer variant, which is the newer strain of the Omicron, how well it's going to target that. And then, again, you see that in the UK population too. The new variants continue to spread. It's that new one. So, again, the hospitalizations are relatively low. This EG.5.1 is to blame for this variant, but the spread. But again, people want to stay safe. People are recommending, in maybe in crowded places, put on your masks again and consider, like, this is a nice article. Should you get your booster before traveling? What they recommend here is, which I think everyone here is obvious for everyone here. If you're immunocompromised or a high risk, yes, you probably potentially get your booster if it's been a few months, like 6 months or longer. But if you're not and you take some precautions inside, then maybe you should wait for the new one to be announced, released, and take the new variant, the new version of the booster. One study that made me chuckle a little bit, but which is good to know too, with the new rising cases. This study that came out in PLOS One, saying that if you pick your nose, you got a higher risk of COVID. So they actually looked at people wearing glasses, bearded or mustached, or nail biting, or picking nose, and you can see from the data. They took a survey, and then they compared with the survey, did like the sort of correlations analysis to people getting infected, and they saw that indeed, if people who picked their nose more had a higher risk of human COVID. So with this new risk coming out, make sure you don't, or wash your hand before you do it. But that's just a kind of a light topic here for the paper that people might be getting humor into that. So I'm creating an other virus news section to go over, just because, with covert, one of the things is not only are we set up to obviously look at COVID, but you could use our tools, knowledge, and there's people have expertise like Nydia, who knows a lot about many different viruses, and other people here also. But just in case, we could use our tools and ideas for future pandemics. So one thing that caught my eye was that monkeypox was predominant was coming up, what, about a year or two ago, a year and a half ago now, I think. But now it seems like in China, obviously monkeypox has continued to decline in the U.S., Europe, and other places. But in China, they've been noticing a spike. I mean, it's low still. You see, it's 117 cases. But one of the things they talk about in this article, getting accurate numbers is difficult. So it might be a little higher, but still, something people are looking at because monkeypox could be contagious by touching, obviously. So this could be something to keep an eye on. But the good thing is, I mean, there's vaccines against monkeypox, but China tends to keep things, from this article, as they pointed out, they keep things more in-house and they are more resistant to getting stuff from outside. One thing I know people brought up is the avian flu. We know that the avian flu here, for example, this article came out a couple of days ago, or a few days ago, talking about Norway and Finland's getting this massive spread in the birds. They already saw like 10,000 dead birds over the past few weeks. And then the UN agencies have been warning that, that we should watch avian flu just in case it might be able to adapt and infect humans. But right now, the risk of infecting humans is very low. I mean, I forget how many it has jumped to humans, I think, but it's only been one or two or less than 10 cases from what I know. I don't know if others have any other thoughts on avian flu, but that's something to keep an eye on, and maybe just for the whole agriculture industry, that's something to maybe people want to dive into using our tools to look at that. And another thing that Diego had brought up, last week, which is something that was interesting, and I looked a little more into it. The risk of, due to global warming and the cases giving, the heat getting higher, these mosquitoes who were primarily carrying, dengue, Zika, or chikungunya, that are primarily in the climates like the Amazon or South American countries that are a lot warmer. They've been starting to see the rise, for example, in Europe, where they're getting their higher heat waves and things, as we know, the past year or two. So they've been starting to report a lot more mosquitoes carrying these things like chikungunya, Zika, dengue, and what's now in the U.S., of course. But so that's something that people might want to look into. And from our South American colleagues, they say that chikungunya is definitely a very bad virus to get and awful, from what it sounds like. So some people want to expand and use their tools, and they have omics data that we should tackle. I mean, I'm more than happy to try to jump on these different ideas because getting ahead of potential outbreaks is what one thing COVID set up to do. But Diego brought up a good point. Yeah, go ahead, Bill.
Do you know what's the degree of cross-reaction in these tests for COVID? For example, when I returned from a DC meeting in late February 2020, I received the diagnosis of avian flu, but they said it could be cross-reactive with the COVID, but since they didn't have a COVID diagnosis at the time.
That is very interesting. I never heard of it.
Yeah, I never heard of it either. I don't know, Nadia. Nadia would be the person I would ask, but if you've never heard of it, then I never heard of it.
Yeah, guys,
I wonder if they do like a multiplex of a bunch of respiratory viruses, and they were unsure which one you had.
That could be it.
Good question. I didn't have the presence of mind to ask what test they were using, but it was quick.
Yeah. But yeah, I mean, that's something I don't know. If Nadia doesn't know the answer, then I'll say definitely it's not commonly known. Sorry, Nadia. I think you're my expert here, so if you don't know it.
Okay, good. Glad to have you aboard, Nadia.
Yeah. All right.
I think Nadia, regarding the bird flu, I just posted an article in the Atlantic that I was recently part of, and I've also been working on flu and avian flu throughout the COVID pandemic. Yeah. And what we are seeing now is this increase in cases in mammals, not so much in humans, but in other mammals, right? Sea lions, seals, cats, mink. And so we don't see in the viral genetics genome like any signals or mutations for adaptation to mammalian hosts. But I think it would be really interesting if people have access to other data, if we could explore this other data to see if we find any evidence of adaptation to mammals.
There was, I think you probably heard about it too. There was one or two cases in the UK, right, that the humans got
Human.
infected with that.
Yeah, but human cases have been reported throughout time. It's usually
Yeah.
around the hundreds, the hundreds in humans. But what we are seeing is the levels in humans continue the same as before, but we are having many more in other
I
non-human
see.
mammals. Yeah.
Hmm.
So, Nydia, this is Kevin, and Ashton already knows this, but I work with an organization, and that's something that I hope that we'll begin investigating soon.
Mhm.
So you'll be welcome as well as others possibly to join that effort, but I'll provide more details in the future, hopefully.
Thank you. Thanks, Kevin.
Yeah, Ken's working on some neat stuff related to zoonotic and other viruses. So.
Yeah.
But yes, okay. Well, if people have other viruses and news to report to keep an eye on, Nydia, maybe you should tell me ahead of time, and I could always put some snippets here of other things that you think are important for us to pay attention to.
Sounds good.
Yeah, I figure that's one of the things I'll start reporting on in these meetings. Other than COVID news, I thought maybe other viruses that we could potentially use our tools we have. And of course, one thing we're obviously going to be focused on is long COVID. Probably people might have seen this article that came out a couple of days ago in Nature, talking about NIH, the Recover program, basically. I know there's been a lot of criticism from it. A lot of people have criticized it because that's what they talk about here also in this Nature article. It's been 2 years, and it's been moving very, very slowly. But some people in the program criticize, clinical trials take a long time, which they do. But what they've just announced was this past week that they're doing their first long COVID human clinical trials on these drugs here. So Paxlovid, everyone here knows what it is, so I'm not going to go into that. But they're testing, if you give Paxlovid longer, 15 or 25 days as opposed to the 5-day regimen that they recommend now, can alleviate COVID symptoms. Which there's a lot of anecdotal evidence that maybe it might be the case. But there's these other 3 drugs, which I thought, which is interesting here, which maybe Doug also has heard of. But I looked at the mitochondrial relevance of these drugs because, as one of our standing theories here, is that long COVID is just a mitochondrial disease. 2 of these drugs that are here that is on a human trial, they're testing for sleep and wakefulness. One of them here is a modafinil. And there's a nice little review about it when I looked at it. Metacomia target organelle directly inhibiting free radical productions to promote ATP for the mitochondria. So, and
Thank you, Mark. Thank you, Ashton.
Yeah. Yeah, I don't know if you have any insight in these drugs, Doug, or have you heard of these?
No, but I'll follow up on it. Let me record this.
There's this other one too, ivabradine, which is used for cardiac type issues, and when you look up what it does, they show that definitely this is involved. So this is a paper. This is the conclusion of the paper in the abstract saying, reduces infarct size independently, a reduction of heart rate, blah blah blah. But it also says this is possibly the mechanism they say in this paper from what they found is reducing mitochondrial ROS formation and increasing ATP. So it has a mitochondrial. There's been papers showing the connection, I should say, from these drugs that they're testing. So they don't talk about in this article anything about mitochondrial or anything how these drugs might impact it. But there's an underlying evidence here that maybe something to do with mitochondria, whether they're paying attention to it or not.
Yeah, our paper coming out next week will be very timely.
Yeah, exactly. So that's something I thought was interesting from reading this article here. There's this new preprint. Again, preprint, take it with a grain of salt, but I thought it was very interesting here because we've been talking about how long does the RNA, the SARS-CoV-2 RNA, persist. I know Rob Schwartz and others here have looked at it that they say, yeah, it's there for a long time, but then is the RNA actually replicating or not? That question is always up for debate. So this preprint, again, this is a preprint, so take it for the grain of salt. What they saw, even 2 years in tissue, so they looked at biopsies, and one thing they looked at, and then they also looked at using imaging, radiopharmaceutical agents like PET and stuff like that, PET imaging. They looked at T cell behavior, T lymphocytes, and then also looked at other factors from biopsies. That's what this is looking at, the proteins here, related to long COVID. and then also quantifying, which I'll show you the figure in a second, the RNA virus itself. Is it in these regions? But they saw basically, this is from the abstract here, from the PET imaging. They saw that there was significantly higher, basically T cell activity happening in these regions: brainstem, spinal cord, whatever, bone, cardia, and so on, and gut wall too, and the cardiopulmonary tissues. And this is from a protein proteomics they did on the post-COVID patients, and this is the long COVID. Here's 5 reporting greater than 5 or less than 5 long COVID symptoms. Is this pink or blue? And here's like 90 days or greater than 90 days. You can read the figure caption here, which was imaged after their symptoms onset. And then the uptake, like how much the uptake for the dye for the PET imaging. So this is interesting. So there's some genes here that probably from the inflammation paper and others here might recognize that are changing for some of these long COVID patients. And if you look at with increasing symptoms, like you see a pattern approaching greater than 5 symptoms, it's kind of a cluster with higher activity of some of these proteins. But this is the figure from the paper that's showing the spike RNA detection for these long COVID patients, basically up to 2 years. So, and you can see this is the different regions. This is from the rectosigmoid tissue. That's what they're looking at here. And they're looking at basically the SARS-CoV-2 spike RNA, which is the green dots. You see here, this is the controls. This is the post-COVID 200 days, 158, 213, 678 days. That was 2 years. But you can see the RNA particles still there, so your control, meaning COVID negative patients. This is from the biopsies they take from the lungs. And then this is looking at the different CD type T cells, different immune, looking at the different comparisons of what's happening with the different time points. But that's interesting to see. They didn't talk about RNA. Again, I read through it quickly, but they didn't talk about if this is replicating. I don't think they said anything about it. I don't think it is replicating, but they claim, potentially, since you see the spike RNA here detected, this could maybe be one of the persisting factors for long COVID and might be correlated with the, higher amount of symptoms, which they were looking at. There's some other figures in this paper too, from the imaging data, PET imaging, looking at T lymphocyte activity. But this is interesting preprint that thought people might be interested to look at. I think their end population, is 24 participants, so it's not very high, but they got significant results from that.
This is very interesting. I wonder if this is something that is just persisting, for a long time, but really not causing infection, or if it's something that in the future can turn into like a mechanism similar to herpes virus, where the virus can be reactivated. I don't know
that sounds like a grant idea, Nydia. Because that's some experiments you might be able to try to do in the lab, right? With reactivation. Yeah, that's it. I don't know. Yeah, that could be. In the herpes virus, is there any of the RNA in there that helps it reactivate, or is this just the recognition of it?
Yeah, that I cannot tell you. I'm not super familiar with herpes mechanisms.
Okay. But yeah, what you mentioned, I think could definitely be evolved into like maybe an R21 as a preliminary grant figures. It can happen, but that's it. Yeah, that's something to worry about, right? 10 years from now, can this reactivate in people who have this?
Yeah.
And they were only looking at 2 years post, because this is what the data have. Is these RNA viral particles, are they in us 10 years from now?
Yeah. If we can do that, like at least show that in laboratory, I think it would be, quite valuable. Mm-hmm.
Again, but this is a preprint, so maybe the data might change, but it seems solid from when you look at the figures. So, any other comments, questions, ideas for research? Right. So I mean, that's the most recent papers that I found that I thought might be interesting to group. I'm sure there's more papers out there. Um, just to give you a little quick update on the variant distribution, we know that EG.5.1, which we talked about last week, is the one that's starting to spread quickly. The wastewater, as we know, you see this uptake here. Nationally, it's showing that again more cases happening, and then places, let's say where I live, I saw this, this is Middlesex, Boston area. We saw the spike going up, but it's coming back down now. So, it's a cyclical pattern. Maybe it's reached this peak and people got infected enough, and the immunity and the vaccination kicked in. I don't know, but let's see this thing happening. It's going back down. So we'll wait till next week to see if this trend keeps going down or not. But nationally, it seems like it's going up, as we've been hearing about reporting. And in the wastewater, we see that EG.5, this mustard yellow color, is creeping up here in the wastewater as it would be expected. The variants in the U.S. again, same. It's the recapitulation, or basically, it mirrors what's happening in the wastewater. From the sequences that CDC is doing, we see EG.15, the yellow one's creeping up, like these other variants did when they take over. And you can see the regions. It's pretty uniformly distributed, the amounts that are there from the west to the east coast. I guess the middle of the country is still a little lower on the EG.1.5 or EG.5, sorry. And globally, like this is all I really have. But again, the case results are not as reliable in most countries. Although in South Korea, they're reporting better in most places. You can see the spike going up. Hospitalizations is still relatively low. Although remember I said earlier, they're reporting increase in hospitalization. Increase meaning in the past few weeks, we have 5% increase. It's still low, which you can see this coming up in the U.S. And this is another version of this from the beginning of the COVID pandemic. But that's it for this week. I mean, next week, Steve had suggested